Good morning. We are here today to present to you our project named Killifish in Space. This was a study of the effects of microgravity on the bone density and muscle mass of these fish, and a test if these fish, being they have kidneys, if they would get kidney stones. I am the principal investigator, Jessica Gunderson. I've just finished my freshman year in high school. These are my co-investigators, Justin Sadler, who just finished their junior year in high school, and Brittany Rigby, who just finished their sophomore year in high school, and Austin Sadler, who just finished their junior year in high school as well. What are killifish? Our genus that we used with the nodobronchius kind. What are killifish exactly? Killifish are small freshwater fish that are located around Europe and Asia. These fish are recorded to grow up to 9 millimeters. Why did we choose killifish? Well, killifish were large enough that they could fit in our we could tell if their bones and muscles had been affected by microgravity, and they were small enough to fit in our FMEs. Another reason was diapause. Diapause is a form of survival. There are 3 stages, 1, 2, and 3. Stage 1 is a form of dormancy that can be up to 100 or more years. Stage 2 can last up to 25. They're getting more or less ready to hatch. Stage 3 is they're ready to hatch. How this works is they get mixed with water to become reactive. Their diet consists, for our purposes, of fish flakes, but in a normal life habitat, they would be feeding off of brine shrimp.
Our hypothesis was that they would have less bone and muscle mass in microgravity. We figured this because, as in humans, they come down from space and they have trouble standing and walking because they have loss of bone and muscle mass. As well as that, humans in the ISS can get kidney stones because as bone degenerates up in microgravity, calcium buildup gets in the kidneys, and we can also tell this on killifish because they have kidneys as well.
For the materials, we chose to use the FME3, or the Fluid Mixing Enclosure Type 3. The experimental and control groups each contain 3 different parts: the main volume, ampule A, and ampule B. The main volume had 6.28 milliliters of spring water in it. Ampule A had 3 killifish eggs and diapause 2 and 2 fish flakes in it. Ampule B was empty because in our original proposal, we were going to separate the eggs and the food. Then it was recommended that we combine them, so we just left that one empty.
For our procedure, after we were selected, we were sent many fish eggs by Dr. Eugene Hall. We had multiple batches of eggs. With our technology at our high school, we were not able to see these, so we took them up to our local university in Storm Lake, Iowa, Buena Vista, and we used their binocular microscopes to do a little more research. We found 3 eggs per tube that looked suitable for this project. Then we took them back and put the materials in this FME. Then they were sent up and docked on the ISS. We requested that they be kept at room temperature, and for this process on the ride home, we asked that a hand warmer be activated. And since we do not have our results back currently, we will be taking up to BVU again for testing to see our results.
While we were at BVU, some of the technology we used was video microscopy, which is you take something microscopic and you can take a picture of it. Here we have pictures of the control experiment on Earth, and you can actually tell they already have organs. Like someplace you can see the eye and the spinal cord that wraps around the backs, and you can see an eye again in that one, and the spine's really prominent in that picture. Here's the photos of our experiment that went on to space, and here you can even see more. You can up to see a heart with the spine. You can see the spine down into here with the heart in the center. And up here you can start to see the eye and the spine wrapping around. We would like to thank the people that helped us with our project, Mrs. Carol Sadler, my mom, and our teacher facilitator, Mr. Jim Christensen, who pulled our school about the project, came and presented that this could happen and that we could be a part of it. Dr. Eugene Hall and the Diapause Foundation that donated all of our killifish eggs, because they are really hard to get a hold of. Dr. Melinda Coogan and Buena Vista University to allow us to use their technology and laboratories to do all of our research in. Jeff Goldstein, program director, to help get this set up. Dr. Rita Fram, she did all the fundraising to send our SSE project into space, and then everybody that did all of our fundraising and that gave us money to allow us to come here. Do we have any questions?
All right, thank you for a wonderful presentation. We are now ready for questions for these researchers. Please come to the microphone.
Hi there. I was wondering, what are you planning? How far do you plan the life cycle of the killifish to include in microgravity, and if you have any plans for them once they come back? Are you going to initiate a full life cycle and run tests on them?
They were up there for 37 days before they were broken and hatched in the water to reduce possibilities of dying from lack of oxygen or food. And we can use these further on as test subjects if they do have kidney stones, because it's been proven that we have kidney stones as humans coming back. So we can use them for further test subjects with other medicines to see if this would help for humans as well.
Thanks.
Let's thank our presenters again.